This is the introductory lecture on nucleic acid structure, and we'll go through nucleic acids and what they're composed of, uh, and we'll also talk about, towards the end of the class, the discovery of the structure of DNA in the cells and how that was made. But the title slide is, I think, a good summary of the four main DNA bases that you'll need to know. You'll need to be able to recognize these, uh, the pyrimidines and the purines, C, T, and A and G. And I like this slide. Not only does it show the structures, but it also has the numbering system for each of these, which is kind of important too when we talk about where these attach to ribose in DNA and RNA, and also when there's a substitution or like a methylation in some of these. We'll see examples of that later in class, where those are in terms of the numbering system will be important. Okay, so today we're going to cover sections 8.1 and then a beginning of 8.2, and that 8.2 is really the discovery of the double helix. Okay. So the functions of nucleic acids and nucleotides, and we'll define those terms here in a second. Nucleotides function in a number of ways. In energy metabolism, they function in energy metabolism, the molecule ATP, and that's something that we really probably don't have many examples of in this class. I suppose active transport we talked about this semester, but when you cover metabolism next semester, that will be a lot more apparent. Okay. Also, another function would be enzyme cofactors, such as NAD plus and NADH that we've talked a little bit about. And then also in signal transduction, which we've covered, in the molecule cyclic AMP would be an example of a nucleotide that functions in signal transduction. Okay. Nucleic acid functions are really, nucleic acids are the polymers of nucleotides, and so their functions are a little bit, I guess, grander in scale. One is the storage of genetic info, that would be DNA, transmission of genetic information, messenger RNA, processing of genetic information, ribozymes. Not sure we'll really cover those yet in this class. That probably comes next semester. And then also protein synthesis with the tRNA, transfer RNA, and rRNA, ribosomal RNA. Okay, so nucleic acids are really in the business of storing and transmission of genetic information. Okay, so what is a gene? That's a good question. Okay. A gene is a section of DNA that encodes either a protein or an RNA. Okay, and so for the most part, this is the only known purpose of DNA. Although there is a lot of DNA that's discovered that really they don't really know what it does, because it's not involved in encoding proteins. So there is a significant amount of that DNA that doesn't seem to have much of a purpose in it, or maybe the purpose is kind of unknown right now. But really the only known purpose of DNA is to encode for protein sequence or RNA sequence. Okay. So let's look at the structures. Okay. Primarily when we're in this class, we'll be talking about nucleotides. When we look at nucleic acids, the really building block of that would be a nucleotide. Okay, so nucleotide has three parts. Okay, the first is a nitrogenous base. And on this figure, we'll just put that in a box here. We won't look at those yet. Okay, the second part is a pentose, right? Pent meaning 5 and ose meaning sugar. Okay, and so the pentose is labeled here. The pentose that is used in these nucleotides is ribose. Okay. And the numbering system is labeled as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime for the carbons. Okay, those prime, they're given the prime after the number because the numbering really starts up here in the nitrogenous base. Okay, so if you would, continue on from that, these aren't, going to be, carbon 1 wouldn't be here. But we call this carbon 1 prime to denote that it's not the first carbon in the entire structure, but it's just saying it's carbon 1 from the ribose. Okay. So those numbers will come up again. Okay. And one of the places they'll come up again is the fact that in DNA, DNA stands for deoxyribonucleic acid. There is no OH group here at the 2' position of the ribose sugar. Okay, so that's why that's labeled pink. Okay. And then finally, the third part of a nucleotide is the phosphate group, which is attached to the 5' carbon of the ribose. Okay, so nitrogenous base, ribose sugar in the ring form, and then a phosphate group. Okay. So that's nucleotide. There's also something called a nucleoside, okay, and that would be the same thing what we just looked at, but no phosphate group. So it only has a nitrogenous base and the ribose. Okay, and then finally, if you're just talking about the nitrogenous base, sometimes those are called nucleobases. Okay, and the two types are shown down here. Pyrimidines are 6-membered rings, and purines are a double ring structure. You have a 6-member ring and a 5-member ring attached. Okay, the numbering system is a little kind of strange. For pyrimidines, the number starts at nitrogen in the bottom here. That's going to be attached to the ribose sugar in a nucleotide. For purines, you'll see the numbering system is a little bit different. 1 is up here on this nitrogen, and then it goes around counterclockwise in that ring, and then 7, 8, 9 are in the second ring. Okay, and purines attach to the ribose via a bond on nitrogen 9. Okay, so that's kind of drastically different how that numbering system works in those. Okay. The phosphate group in a nucleotide, it's going to be negatively charged at a neutral pH typically found in cells. Okay, and again, it's attached to the 5' position, carbon at 5' position of the ribose. Okay. Nucleic acids are built using 5' triphosphates, so they have not just one phosphate group on them, but 3. And those would be designated as ATP, stands for adenosine triphosphate, GTP, TTP, and CTP. Right? Those are the 4 bases of DNA. Right. Nucleic acids, when they are built in chains, they have one phosphate group per nucleotide in that chain. Okay. There's also phosphate groups can be attached to other positions. Right. As I mentioned, the pentose in nucleotides, the 5-carbon sugar, is ribose. And in particular, it's beta-D ribofuranose in RNA and beta-2'-deoxy-D ribofuranose in DNA. Okay, and so that just means the furanose means it's in the 5-member ring form. And beta, remember beta means that the OH group is at the carbon 1, the anomeric carbon, is going to be pointing up. Right, so and this is where the nitrogenous base is going to attach at this position. Okay, so they're when we draw these pictures of a nucleotide, that base, it's going to be pointing up from there, from that position. And then how this ring forms, it's forming between carbon 4, the oxygen on carbon 4 is attacking this carbon 1 with the carbonyl, right, to make that 5-membered ring. So what are the bases? So all the bases are going to be derivatives of pyrimidine or purine. Okay, they're weakly basic because of the lone pairs on nitrogen, and they all have nitrogen in them. That's why we call them nitrogenous bases. Okay, they're weakly basic and they contain nitrogen. They're fairly planar. You can think of them as being planar structures. Hydrophobic at neutral pH. And another thing about them that's important, at least in a laboratory setting, is that they do absorb UV light. Okay, somewhere between 250, 270 is kind of their maximum absorbance of UV light. And so the 5 that are the most important to know, because they're the nitrogenous bases in DNA and RNA, are cytosine, thymine, uracil, adenine, and guanine. Okay. Cytosine, thymine, and uracil are pyrimidines, and adenine and guanine are purines. And we'll look at the structures in some more detail. Okay, so let's look at the pyrimidine bases. Okay, cytosine is abbreviated as a C, capital C. It's found in both DNA and RNA. Thymine, abbreviated with a T, is found only in DNA, and uracil U is found only in RNA. Okay, so T is replaced by U in RNA. All are good H-bond donors and acceptors. Okay, and that's, as we'll see, that's how these interact in the DNA double helix. Okay, and all are neutral molecules at pH 7. Okay, so if we look at the structures in some more detail, right, where to start? Probably, I don't know, cytosine is, well, uracil is definitely the simplest, because you have two carbonyl groups, one at carbon 4, this would be carbon 4, this is carbon 6. Okay, so, and you can remember the numbering system is shown here, and I kind of misspoke there. So by this numbering system, it would appear that this would be carbon 2, that's carbon 4. Okay, so their uracil is pretty simple, I guess, to remember that. You have two carbonyl groups. Okay, cytosine is very similar to uracil, except that the carbonyl at carbon 4 is now replaced with an amino group. Okay. And then thymine gets a little bit more complicated. It's like uracil, but it just has been methylated, I guess you could say, at carbon 5. Okay. So these are the 3 important pyrimidine structures you would need to be able to recognize. Okay. Purines, there's only 2 purines, adenine, abbreviated as A, and guanine, abbreviated as G. They're both found in RNA and DNA. These are also good H-bond donors and acceptors, and they're also neutral at pH 7. Okay. Adenine is the simplest. Okay, it's just the basic purine double ring structure with a nitrogen, one amino group up here. A guanine, a little bit more complicated. Instead of that amino group, you have a carbonyl, and then you have an amino group at the carbon down here. And the numbering system, what was that? That would be carbon 2, I believe, because I think it goes 1, 2, 3, 4, 5, 6, if I remember correctly. And this would be carbon 6 at the top. So the adenine amino group at carbon 6, guanine amino group at carbon 2, and carbonyl at carbon 6. Okay. So these nitrogenous bases attach to a ribose sugar in the nucleotides via a glycosidic bond. And we call this a beta N glycosidic bond. And that's because it's in the beta position with respect to ribose. Okay, so remember alpha would be pointing down, but this is pointing up, so this is beta. It's in the cis conformation with the CH2 group here. Okay, so beta, and it's an N-glycosidic bond because it's not a normal glycosidic bond that you'd have between this carbon and an oxygen. This carbon's attaching to a nitrogen in the nitrogenous base. And again, in pyrimidines, it attaches to N1, and in purines, it attaches to N9. This bond is really pretty stable. It can be cleaved by an acid treatment, but in general, that bond between the ribose and the nitrogenous base is really stable. The same can't really be said for the phosphate groups, especially if you have a triphosphate. So the conformation around this bond can freely rotate. So the base, the nitrogenous base, can kind of be swinging around, rotating. Okay, so how do you know what conformation's in? Well, there's a torsional angle that's defined between the oxygen here and the ring, the ribose ring, this carbon, and then either this nitrogen and this carbon in a purine, or it's defined from this oxygen, this carbon, this nitrogen, and C2 of a pyrimidine. Okay, so that would be C2. Okay, that angle, and there's a couple terms that we're going to talk about conformations in nucleotides with the relative position of the base. So one is the syn conformation, and that's where this angle here is 0 degrees. So basically with 0 degrees, what does that mean? That means this bond and this bond are parallel. They're sort of overlapping on top of each other, pointing the same direction. Okay. That's called the syn conformation. Okay, the anti-conformation is the opposite. It's where you have this bond and this bond would be, going different directions, so 180 degrees. So this one would be pointing that way, and this one would be pointing this way. Okay. In normal DNA, it's the anti-conformation is what occurs. Okay. Don't really know if we see many examples of this conformation, but especially the syn conformation. Sometimes you see them drawn that way, I think just because it saves space on a page to draw it like this as opposed to like that. But in DNA, it'll take this conformation. Okay. Nitrogenous bases absorb UV light, like we mentioned, and again, that range is somewhere between 250 and 270, where they're maximum. Okay? And that's due to pi to pi star electronic transitions. And if you look at these molecules, right, they have lots of double bonds in them, so there's lots of pi electron orbitals for this to happen. Okay, so typically what happens, a molecule like that will absorb a UV light. An electron will go into a excited state, and then that excited state will usually, in some molecules anyways, that excited state will emit another photon to relax back down to the ground state. And that photon that's given off, that's called fluorescence. Okay? DNA doesn't do that. Okay? And that's because there's a lot of what's known as radiationless transitions. Um, and those are from vibrations in the molecular bonds and in that sort of thing in degrees of freedom in the molecule. So they don't go undergo fluorescence, but they do absorb UV light. And so the absorption of UV light, and then the fact that they also have this radiationless transition that relaxes back down to the ground state, actually is a really good effective means of protecting genetic material from UV light. Okay, although UV light is, as we know, is still damaging to DNA. You can use the absorption numbers for DNA and RNA to tell how pure it is. You can measure concentration by it, and you can tell how pure it is. And the way you do the purity measurements is that you do a ratio of the absorption at 260 nanometers divided by the absorbance at 280 nanometers. Okay, and for DNA, the pure DNA, that ratio is going to be about 1.8. For pure RNA, that ratio will be about 2. Okay, so that's sort of a trick in the lab that you use to determine how pure if you have a sample of DNA, how pure it is. Summary of some nomenclature to remember. Okay, we have the 5 bases, 2 of them purines, adenine and guanine, and 3 pyrimidines, cytosine, thymine, and uracil. Okay, so those are just the bases. The nucleoside versions of those we would call adenosine or deoxyadenosine if it's in DNA, guanosine, deoxyguanosine, cytidine and deoxycytidine, thymidine and deoxythymidine, and uridine. Okay, those are nucleosides. Okay, the nucleotide takes a slightly different form. Okay, it goes from something like adenosine in the nucleoside to adenylate. So that YL8, the illate part of that's telling you that there's a phosphate or phosphate groups attached. Okay, so adenylate, deoxyadenylate, guanylate, deoxyguanylate, ceditylate and deoxyceditylate, thymidylate and deoxythymidylate, and uridylate. Those are a little bit difficult for me to say. But those would denote the nucleotide form. Okay. So now let's look at our deoxyribonucleotides, so DNA. These are our DNA bases. Okay, you need to know the structures, names, and symbols, both the 2-letter and 3-letter symbols for these. Okay. The nucleotide names given here at the top, and also in parentheses a little bit maybe more descriptive name for it, right? Deoxyadenosine would be the part shaded pink, the nucleoside name, and then you add 5' monophosphate to tell you exactly how many phosphates are there and where the phosphate is. So you can either call it deoxyadenylate or deoxyadenosine 5' monophosphate. Okay. The symbols, A, you can use an A. That doesn't really tell you that you're talking about DNA though. So probably a little bit better symbol to use would be the lowercase d-a. That tells you that's the deoxy form of this. And I should point that out too. See, at the 2' carbon, that there's no OH group there, it's a hydrogen. So that's why this is deoxy. Or you can use, if you're talking about the entire nucleotide, lowercase d amp. Okay. So those are the 4 deoxyribonucleotides in DNA. Okay. The ribonucleotides are also important to know. They change based on the fact that there's the only difference between these and these is that there is an OH group at the 2' position. Okay, so RNA, these are ribonucleotides, which is RNA. So they have that OH group, and the deoxyribonucleotides, DNA, does not. Okay, everything else in these structures are similar. Okay, except the fact that you have T is your DNA base and U is the corresponding RNA base. Okay, and so their naming system is also given there. Okay, RNA is a little bit more diverse than DNA. DNA really just stores genetic information. RNA can have a few different functions, and some of the examples we will give here are the fact you have ribosomal RNA that makes up ribosomes. You can have messenger RNA that sends the genetic information from the DNA to the ribosomes, and then you have the transfer RNA that converts messenger RNA to protein at the ribosomes. So there's a little bit more diversity in RNA. In RNA, you can actually have some RNA molecules that can catalyze reactions. Okay, so it's thought that RNA was really the first biomolecule, or at least maybe the precursor molecule to DNA, to be able to store genetic information and pass it on or make copies of itself. There are also some what we call minor bases in DNA that are, not the primary 4 bases of DNA that makes up your DNA sequence, but some of them are, for instance, are based on those bases, but they're just changed, altered. And one way of altering or modifying these is by methylating them, adding methyl groups. Okay, and that methyl group is added after DNA synthesis. And it's thought to be what's known as epigenetic marker. Okay, it's a way to mark, in bacteria cells, it's a way to mark the bacteria's own DNA so that the cells can degrade foreign DNA. It's also a way to mark which genes should be turned on or should become active in eukaryotes. And this field of epigenetics is really somewhat new and expanding pretty rapidly. And so that's the study of, does your environment affect your DNA, and does it affect your DNA in a way that is you're able to pass that on to the next generation? And one of the ways it could be infected is by methylating certain DNA. So let's look at some of these. So cytidine C, if it's methylated at the 5' position, or shouldn't say prime, the carbon 5 position, that would look like this. Okay, so that's one of these methylated bases of C. A methylated version of A is shown here, right? And that occurs at the nitrogen that's at carbon 6, so we call this N6 methylated. Okay. You can also have another component or alteration of C, and that's where you have a hydroxymethyl group, so a methyl group with an alcohol attached to it at that carbon 5 position. Okay, and then also N2 guanosine, which would be G. You can have an N2 a methyl group on this N2 that's on guanine. Okay, so that's some ways in which these can be methylated. Okay, some other minor bases that are important. Inosine. Okay, inosine is another base that we need to know, and it's a purine. Okay, and inosine is very closely related to guanine, except guanine that you have that amino group taken off. Okay, so you only have one carbonyl there. Okay, so in that way it looks like adenine, but instead of an amino group, it's a carbonyl group. Okay. Inosine is found sometimes in what's known as the wobble position of tRNA, and you'll talk about what that means, wobble position, next semester. But it's really the last a codon would be three nucleotides, and the wobble position would be the third position in them. And again, it's made by deaminating adenosine. Okay, so that's probably another thing that it might be a little confusing, because it looks like guanine, you can just take off that amino group, but how it's actually synthesized in the cell is that this amino group is replaced with an oxygen in adenine. And this just provides it's thought to provide, another kind of wrinkle into the genetic code, make it a more richer code. Okay. The other minor base that we'll talk about is pseudouridine, and that sometimes is abbreviated with that Greek letter psi. This is found in tRNA and ribosomal RNA. Okay. Where are you? There you are. Okay. So this guy, the difference here, it's quite striking. It's the same nitrogenous base. It'd be uracil, right? That's why it's called pseudouracil. But instead of the ribose being attached to nitrogen 1, it's attached to carbon 5. Okay, so that is just a different attaching point for the ribose. Okay. And then we have some others down here that may or may not see examples of in the class. Probably not. The more important ones to remember would be the methylated versions here, and then these two minor bases. And will they come up again in this class? Probably not a whole lot, but when you go on to next semester, that's when you'll see these examples in terms of where they're important in transcription or translation of genes, genetic material. Okay. Well, you can have nucleotides, by themselves, but as we've mentioned before, they can polymerize and form what are known as polynucleotides, or just in general, that would be the term nucleic acids. A covalent bond is created between nucleotides by what's known as a phosphodiester linkage. So the phosphate group at the 5' position on one nucleotide will bond with the 3' carbon on an adjacent nucleotide. And so that's why you get this term the 3 prime, if you do this, make a chain like this, one side will have an open 3 prime end where you have that hydroxyl there. The other side will have an open 5 prime end with the phosphate group. So that makes these polynucleotide chains have ends to them, specific ends. There's a direction there. Okay. And just by convention, we always read a sequence of a polynucleotide in the 5 prime to 3 prime direction. Okay, so it always goes 5 prime to 3 prime. Okay. This bond here, this phosphodiester bond, is actually pretty stable, and that's one of the reasons why they think RNA evolved into DNA, because missing this OH group here at the 2 prime position of ribose really makes this phosphodiester bond pretty stable. And the example the book gives for the stability here is that scientists have been able to isolate DNA from mammoths that have been dead for long periods of time, and I think have even tried to clone them using that DNA. They've at least been able to sequence that DNA. Okay, so that these are very stable. The bond here can be cut, and that there's special enzymes that do that are called DNase that cleave that bond. Okay. The RNA backbone, as I mentioned, sort of alluded to, is not very stable at all. Okay, when you're working with RNA in a lab, you have to be much more careful about your handling of it so you don't damage it. Okay, the book says it's in water, it will last for a few years, but in reality, like when you're working in a lab, you really have to be extra careful with the water you're using, the tips and sort of plastics. You have to be absolutely sure there's no RNase molecules in there, enzymes that will break it down further. And the reason, we'll see a better example of this on the next slide, but having that OH group there in close proximity to the phosphodiester bond is troublesome for the stability. DNA and RNA are both linear polymers, meaning there's no branching, there's no cross-linking in them. This is a cartoon picture of if we had a chain of nucleotides. This would be, the depiction here is you have the phosphate group at the 5 prime end. This would be the ribose, and this is the base, the nitrogenous base. Okay, the ribose at 3 prime is attached to a phosphate that goes to a 5 prime, and that chain continues. Okay, if you wanted to read the sequence of this, then you don't focus on the phosphates and the riboses, because those are all going to be the same, right? You're just going to read the sequence A, C, G, T, A. And you go from the 5 prime to the 3 prime. So sometimes you might see this written as 5 prime to let you know that this is the 5 prime end. And then you have your sequence of nitrogenous bases followed by 3 prime at the end. Again, by convention, that's how it's normally done. So you can even sometimes omit those 5 primes and 3 primes and just write it out as the letters of the bases. Okay, all of these really mean the same thing. RNA, again, as I've said, it's unstable, particularly under alkaline conditions. And there's enzymes, RNases, that can cleave this bond as well, and they're happen to be fairly prevalent, so that's why handling RNA in a lab you have to be pretty careful. Okay, there's this is just for your information, kind of. I wouldn't test you on these, but these are different types of RNases that are around. and what they do, why they're around. For one, this plant RNase, it prevents inbreeding in the plant, which is a good thing, because if you have a lot of inbreeding going on, that's gonna affect the progeny, and the progeny will be susceptible to certain poor genetics, can really play a part if there's a lot of inbreeding going on. Okay, what happens? Why this bond is not very stable is shown here. Especially under basic conditions, this proton can be abstracted by an OH group. Okay, and when that happens, the electrons from this oxygen will attack the phosphate and cleave this part of the phosphodiester bond. Okay, so you get sort of a cyclic phosphate derivative here on this side, and then an OH group at the 5' end on the next base. But what that really does, the important part is it's cutting that bond, so these are no longer these two parts of the chain would be no longer connected. Okay. Oh, and I should mention dicer. It's an enzyme that cleaves double-stranded RNA. Okay, and it's thought to be good for protection from viral genomes, and it's used in what's known as RNA interference, or RNAi technology, biotechnology, which is used in laboratory settings to silence genes. So you silence a gene, and you see what effect that gene had on the overall phenotype of a cell that you're working with, or sometimes even in animals. Right, so the remainder of the class will sort of go through the road to the double helix. I think we all know already that DNA structure is a double helix, but I think the historical buildup to this is important to discuss. It has been called one of the most important discoveries in biology. The determination that the structure of DNA is a double helix. Why is it important? Well, it finally connected genetics and chemistry together. Okay. Before that, there was really no exact chemical reasoning for in terms of genetics, why things passed on how they passed on their information, and how they stored their genetic information, how organisms did this. And this really was kind of the link. And it paved the way for modern medicine and also modern biochemistry. There's so much medicine going on today that really is centered on genetics and this idea of personalized medicine. How easy it is now, relatively, to get a sequence from a patient, a DNA sequence, and be able to make medical diagnoses, or the medical treatment options based on genetic information. It's really light years ahead of where it was, even 10 years ago. Right. It's also this looking at the historical path to the double helix is also a good illustration of science in action. Hey, there's missteps along the path to a discovery. It highlights the value of knowledge, the value of collaboration, and it also showed the cost of sharing data too early, unfortunately for some. Okay. So the covalent structure of DNA, there was people have been working on this for a while. Friedrich Miescher was the first one given credit, at least, that really, for isolating DNA, and he called this nuclein from cell nuclei. And people were experimenting with it. They would do hydrolysis, with acid, break this nuclein down with acid, and they found there's phosphate groups, there's pentose, and there's a nucleobase. So they knew there was three components of it. They knew the ratios of the three components, and they also would do chemical analyses, and they would find there was phosphodiester linkages. So they knew that the phosphate groups were involved in the linkages, and that the pentose was ribose sugar. Right. So in 1929, Levine in London published this structure. Yeah, not too far off, really, I guess. You'd have the base, and then that was connected to your sugar, and then there was also this phosphodiester linkage. Okay, not quite right. Okay, they just had these linear type sugar molecules here, but not all that far off either. Okay, and then in 1935, Levine and Tipson published a slightly different structure, and so this one was actually, pretty close, right? It is the structure that we see. They determined that the phosphodiester linkages were coming from the 5' of one ribose to the 3' of another, and that the nucleobases were at the 1' position, attached to the 1' position of the ribose. Okay, so that was really 1935 was when the chemical structure of DNA was really solved or determined. I guess determined is kind of a bad term, but the correct, what we would consider the correct chemical bonding in DNA was published. Okay. Well, there was other experiments, so in 1935, it was still sort of unclear if DNA was actually an important molecule for inheritance, and nobody really knew what it did. You could tell that there was a lot of it inside of a cell, inside of the cell nucleus, but no one really knew what it did. And so there were some experiments that led to that determination. Okay, one of them was using bacteria, and in particular, it was this Streptococcus pneumoniae, which is the bacteria that causes pneumonia. There's two strains of it, a rough colony R and a smooth colony S. And the rough colony is benign. Okay, it doesn't cause an infection. It lacks the protective capsule around it. So it's recognized very easily by the host immune system and destroyed. And the S strain is the one that's virulent. It has this polysaccharide capsule around it that prevents immune detection. Okay. So Avery, McLeod, and McCarty did this experiment in 1944, and there was also the Griffith experiment in 1928. Similar. Okay, so using live encapsulated virulent bacteria, okay, remember that's the S colony, smooth. They would inject that into the mouse, and the mouse would die. Okay, and when you did the rough or R strain, and those are the non-encapsulated. Okay, these are still living, they just don't have that capsule around them. You inject that into a mouse, and the mouse lives. Okay, so those would be the controls of the experiment. One is the positive control you can think of, injecting this virulent bacteria, the mouse dies, and a negative control, you inject the non-virulent bacteria, the mouse lives. Okay, so then they did some combinations of these. Okay, they would take live virulent bacteria with that are encapsulated. They would heat them up to kill them, and then they would inject them, and the mouse would live, right? The bacteria now is dead, so the mouse lives. Okay. Then they did a kind of an interesting experiment, and this is in specifically Griffith's experiment of 1928. Okay, he killed the bacteria, some of it, and put a small amount of that bacteria that had been killed by heating it up into some live non-virulent bacteria culture. Okay, and then he mixed those and let them culture together for a certain amount of time. And so injecting either of these by themselves, the mouse would live. But when he mixed them together, the mouse would die. Okay, so what does that show? Okay, alone, heat-killed virulent does not kill. When mixed with live non-virulent bacteria, it does. So the conclusion that was reached is that one type of bacteria can transform another that is normally not deadly. Okay. So this transformation is a term that we still use today whenever we add bacteria in the form of like a plasmid to whenever add DNA in the form of a plasmid to bacteria, we call this a transformation. Right. So again, this experiment, pretty important experiment. The conclusion reached was that something in this bacteria that's when it's living will kill the mouse. Something in these ones that have been the bacteria that have been heat inactivated transfers and transforms these non-virulent bacteria into virulent bacteria. Turns out that that's DNA, but that discovery was a little bit further down the line. Okay, and so what molecules are involved in this? Okay, so you have to figure out how to isolate the different components here. Okay, proteases destroyed proteins. The bacteria was still deadly, right? So you would put proteases in with the virulent bacteria. Okay, proteins would be destroyed, but the bacteria was still deadly. They tried to remove lipids and carbohydrates from the bacteria, but the bacteria was still deadly. Okay, what was left? Well, the only thing left, right? You're looking at proteins, lipids, and carbohydrates. Really, the only thing left in terms of a biological molecule is DNA, nucleic acids. Okay, then they use the enzyme that destroys DNA, DNase, and the bacteria was not deadly. Okay, so that really answered the question, how is this happening? Well, it's happening because there's transferring of DNA from one, in this case, the heat inactivated virulent form is transferring DNA to the non-virulent form. And then they went a step further, right? Now they're adding the live non-virulent bacteria and then just DNA isolated from heat-killed virulent bacteria. And that's when they would see this transformation, and they'd actually get the bacteria to start forming those capsules and being encapsulated, right? And as you'd expect, when that was injected, the mouse would die. Okay. So the conclusion at the time, DNA contains inheritable characteristics. Okay. Avery cautiously suggested that DNA was the genetic material, suggested that this could be true in higher organisms as well, right? Because they're just looking at bacteria. There's really no good way of doing that same sort of experiment in a human. Okay, so you just have to make kind of an inference there. Okay. This was the first experimental evidence that DNA is genetic material. Okay, at the time, it seems pretty obvious to us right now, but at the time, it was really controversial, and not well received. The current thinking at that time was that proteins were where the genetic material, the inheritable characteristics came from. Right? And DNA was just something that held proteins, the important proteins for that together in chromosomes, but didn't really do, but the DNA didn't do anything. The proteins did everything. Okay, which is not the case as we know now. Right. In 1952, so those experiments were mid 30s to early 40s. The early 50s saw some other experiments, some really kind of this field sort of take off here. Um, Hershey and Chase in 52 wanted to really prove this idea that DNA was the inheritable material. That's where it was coming from, and so they used viruses. So they're trying to determine, okay, what is transmitted from a virus to a host, and the conclusion was that the viruses transmit DNA, not protein, and this was really like the final proof that DNA was the genetic material. Okay, how did they do this? Well, they used radioactivity. They would label some viruses with radioactive phosphorus, so P32. Okay, all phosphorus is in DNA, in a virus. So all phosphorus in DNA is in all phosphorus in a virus is in DNA. So the protein in a virus kind of makes up the capsule or capsid, and then the DNA is inside. So in the P32 experiment, you had radioactive DNA inside the virus in the DNA. They'd also labeled some viruses with S35, sulfur 35, which is radioactive, and that all sulfur is in the protein of the virus, the coat protein. Right? Remember, proteins have cysteines and threonines, amino acids in them, so those would have the sulfur groups that are radioactive. In DNA, you have those phosphate groups; those would be radioactive. Okay, so what they saw when they let those, and these are what are known as bacteriophages. They're viruses that infect bacteria cells. So what they saw when they did both of these experiments, in the P32 experiment, after they infected the bacteria with the virus, they noticed that there was radioactivity inside the bacteria cell. I'm kind of jumping ahead here. That's on the next slide. Okay, so the viruses would infect the cells, and then they would treat the cells so that the remaining pieces of the virus would be taken away. Okay, and at the end of this, what they saw is that the viruses that had treated with radioactive phosphate transferred that into the cell, and these cells now were radioactive. The viruses that had the S35 in their protein coat, the cells that were infected with those viruses did not have any radioactivity in them. So that really proved once and for all DNA is what is the genetic material that's being transferred by a virus into the host cell to change its genome. Okay, so DNA, not protein, carries the information needed for the replication in a host cell. Okay, after that, it was well accepted that DNA is the genetic material. Okay. Okay. Some other important experiments came from a person known as Chargaff. So Chargaff's experiment showed the typical makeup of DNA, and he looked at different sources of DNA, and then he took what percentage of that DNA is, certain bases, okay, the 4 bases. And that really didn't show a whole lot. Sure, some of these are similar, but they're all, kind of different. What was probably the better step that he did was he actually found ratios of A to T and G to C, and he noticed that those were very close to 1-to-1 ratios. Okay, and so that was really one of the big take-home points of his experiments was there's 1-to-1 ratios of A and T and G and C. And he came up with, and this is from the 40s, and he came up with what are known as Chargaff's rules. Okay, one, base composition of DNA varies between species. Okay, so and also the percentage GC that's varying between species. DNA from different tissues of the same species has the same base composition. Okay, so when you look at DNA from your skin and your a muscle, let's say a muscle sample, that those are going to have the same DNA makeup in terms of, as we know now, the sequencing. But he was really just looking at the makeup of the bases, the base percentages. Okay. Base composition of a species does not change as the organism ages or with health concerns or nourishment. So this base composition is not changing over the lifespan of an organism. And regardless of species, the amount of A equals the amount of T, and the amount of G equals the amount of C. Therefore, purines equals pyrimidines, or A plus G equals T plus C. Okay, so those are the Chargaff's rules. Okay, and this slide details, we're going to talk about this slide maybe in a little bit more detail at the start of next lecture, so I won't go into really all the details, but it was in the 50s, the early 50s, that the actual structure of DNA was determined, and that was by X-ray diffraction. So that was around the time that people were using X-rays to look at molecular structures quite a bit. The structure of alpha helix was determined very, very soon after this. I believe it was right after this. So this was really the time when this was becoming big. Okay, it was found that it was helical. This diffraction pattern does not show a double helix. Okay, it just shows that it is helical, and it gives some dimensions to that the helix. Okay, so that was determined by Rosalind Franklin and Maurice Wallace. They did that work. Watson and Crick really were the ones that came up with the idea of the double helix, and that's because they took the work that was done by Rosalind Franklin and Wilkins and put it together with Chargaff's rules and came up with this idea of an anti-parallel double helix, and it fit the data. Okay. Watson and Crick and Wilkins shared the Nobel Prize in 1962, but unfortunately, Rosalind Franklin had died before then. I'm not sure whether she would have shared in it or not if she was alive. But once you die, you can't win a Nobel Prize. They don't award those after death, so which is really unfortunate. Okay. So the interactions in the double helix are, at least the interactions between two strands of the double helix, involve hydrogen bonding. Okay. Two bases can hydrogen bond to form a base pair. Okay, for monomers, a large number of base pairs is possible, but in polynucleotides, only a few possibilities exist. And so Watson and Crick determined that the double-stranded DNA A pairs with T and G pairs with C. So they use the Chargraph's rules to kind of come up with that. And and you can also think of that as purine pairs with pyrimidine. Okay, we'll look at these pictures probably a little bit more in detail next lecture. Okay. So what we have with the Watson and Crick model, it's anti-parallel, meaning one strand is running 5' to 3', and the other strand would be running 3' to 5'. Hydrogen bonding is very important in this model between the strands, and then you have what's known as base stacking going on within the same strand. So bases are sort of stacking on top of each other as you move down this helix. Okay. They're complementary sequences, meaning their nucleotides are matching A with T and G with C. The absence of the 2' hydroxyl groups allows bases to lie perpendicular to the helical axis. So the helical axis would be going this way. The bases are perpendicular to that, right? So they're kind of, you can think of them as flat or horizontal here in the vertical helix plane. So next time we'll pick up here again. We'll kind of go over that a little bit in review, and then we'll finish the chapter going through section 8.3, dealing nucleic acid chemistry, and then the last section of chapter 8, other functions of nucleotides, and that's sort of what we mentioned kind of at the beginning of class, how you can get chemical energy from them or use them as enzyme cofactors or regulatory molecules.